Welcome back to another session of regeneration biology. And today we will learn about how different polarity or different signaling events that decide the gradient or polarity of in the animal's body and gradient or polarity of a given chemical, how it is contributing to the regeneration. So if you look, this is the plenary anatomy, as you can see here in this picture. And it's very complex anatomy in the sense, although it's a flatworm, it has got eye spot, which is basically the light sensor, and also it has got a cerebral ganglia, and it has got an extensive peripheral nervous system which is marked, and also it has got intestine, and also it has got a pharynx which is hanging out, that is the mouth and the anus. It has got the mouth and anus, same thing, doesn't have any dedicated mouth or dedicated anus. And so it also has transverse nerve, and also it has got dedicated secretory cells which is distributed throughout its body, means nitrogenous waste material, which is normally called as flame cells. And this picture is mainly shown for you to understand that the animal has got a very complex body structure. It is not as simple as hydra, and still it can regenerate by something called fragmentation, or you cut into as many pieces as you want. The animal regenerates from each piece. So this is the planaria anatomy. So now the interesting question, which cells formed the new head or tail? Although we have seen they have got neoblasts, and we call them as clonogenic neoblasts, neoblasts. But the question is, although they are there, they are circulating, which one? Is it throughout each of them? All of them have got equally potent? Any given cell give rise to any given tissue type? Or are there any predetermined distribution of function or labor distribution is there? For many years, it was believed that the old cells dedifferentiated at the cut ends of the planaria to form a regeneration blastoma. Dedifferentiation, that is going back in time. Like I gave example probably in the previous class, someone who is studying in the +2 is now going and sitting in the kindergarten. So this is something dedifferentiation. That was the belief olden times. A collection of relatively undifferentiated cells that can be organized into new structures by paracrine factors located at the wound site. This was the belief. At the injury site, there is a lot of differentiated cells. They dedifferentiate and give rise to a naive type of cells which can give rise to the lost structure. This was the belief. However, in 2011, a series of experiments by Wagner and colleagues provided evidence that dedifferentiation does not occur. There is nothing called a dedifferentiation exists. Until then, that was the popular belief, because there is a formation of a blastoma. And how come that cells formed? From where it is coming? The animal doesn't have that group of cells. So the belief was it dedifferentiated at that site. But later it is proven it is not correct. The regeneration blastoma forms from pluripotent stem cells. They are called clonogenic neoblasts. So this is the one of the earlier learning of how regeneration occurred in planaria. A set of pluripotent stem cells in the flatworms that serves as stem cells to replace the aging cells of the adult body. Any given body like our skin has got lot of shedding and the replacement. Whether you go sunlight or whether you have wear and tear, no matter what, your epidermal cells do die and they grow back because of the skin stem cell. Adult stem cell, remember we studied, and the skin stem cells replace them. But same process is happening for planaria, not just for skin, but for every tissue, every organ in its body, and which is done by C neoblast. They have a cell surface marker. C neoblasts have a cell surface marker called tetraspanin 1. This is the name of the protein that acts as a marker for this C neoblast. The picture shows what you see. The previous slide also, this picture was there, and this picture shows that the neoblast in the planaria flatworm, and this is Smithia mediterranea. In short form, it is called SMED. Stained red by antibodies to phosphohistone 3. Phosphohistone 3 is a marker for labeling cells currently undergoing mitosis. Phosphohistone 3 will mark only those cells which is currently in the mitotic phase, means a cell is dividing into two. Not those cells which are in cell cycle. Maybe if there are 100 cells which are in cell cycle, but some may be in G1 phase, some may be in S phase, some may be in G2 phase, but this will mark only those in the M phase. And if you stain with phosphohistone 3, as you can see here in this picture, they are stained in red in color. You can see how much of them are in the mitotic phase. A huge percentage is in the mitotic phase, and remember, this is a normal planaria, no injury. And all nuclei are marked in blue. That means the rest of the cells in the body, they are marked in blue. The one which are red in color are the one which are marked with the phosphohistone 3. Neoblasts are scattered throughout the body posterior to the eyes. That is, where the eyes are there, below the eye is the main location of the neoblast. Assuming like you think planaria like something like that of human, when you stand up, you have two eyes. Below the eye is the place, major distribution site for neoblast. And they are scattered throughout the body, but the preference is below the eye, posterior to the eyes, but are absent from the centrally located pharynx. The centrally hanging pharynx is there, through which it acts as both mouth and anus. There is no neoblast in them. That is the only exceptional area. This doesn't mean that if pharynx is amputated, it will not grow. Nothing like that. It can grow, but they are not used as a reservoir of neoblasts. So in this experiment, what this Wagner and colleagues, what they did was, they performed some very interesting experiment. So this is the picture which we have seen. Neoblasts are there. So they did two different kinds of radiation regime. They took this normal SMED type of SMED planaria and irradiated. And one set they gave 1750 radians, 1750 radians only. What is the beauty of this radiation? It damages almost all. It do not damage everyone, almost all. Maybe some 3 cells will survive, or maybe some 5 cells will survive. This is the idea. In another set, what they did, they gave 6000 radians. That means forget about having any live cells in that animal. It's basically a skeleton, or basically a dead body of planaria. So this is the 2 sets. What they did, they wanted to know, is there any clonogenic neoblast? What we know now is that there are something called neoblast, which we call it as clonogenic neoblast. But how do we prove that? Or you cannot just name them and say that, oh, you from today onwards, you are clonogenic neoblast. There need to have some evidence. Clonogenic means they can create a clone of one individual. Like if one cell from my body can give rise to clone of myself, then I can tell, oh, that cell is a clonogenic cell. That means one cell can give rise to the clone of an organism. So Wagner and colleagues showed that if a planaria are irradiated 1750 radians, which kills nearly all dividing cells, that is neoblast, keeping a few or one alive. That is the radiation. It is lethal, but not absolutely lethal for every cell. Some cells do survive. This would be sufficient to produce the cells of all germ layers, indicating that they are indeed pluripotent stem cells. Say after radiation, 1750 radiation, you leave the planaria as it is. Normally it should die. Like we have in the previous class also, we have seen if the radiation is very severe, it will become disintegrate. But 1750 radiation, if you give, it bounce back. But it had only 1 cell, 2 cell, or maybe 3 cell of neoblast. But that is good enough to repopulate rest of the body. Next, the same researchers, they irradiated the planaria with 6000 radians, which killed all neoblasts. Technically, that body will disintegrate. These planaria died because of failed tissue replacement. No point what you do. However, the introduction of a single clonogenic neoblast into such worms could, in some case, restore all the cells of the organism. Sometimes all will not be successful, but one is enough. Although you have treated them with 6000 radians, one is good enough to restore the entire organism, which is equivalent to the genotype of the donor C. neoblast. When this worm was split into pieces, each piece regenerated the missing parts and the cells were all of the genotype of the donor neoblast. Naturally, the 6000 radiance of radiation has killed all the host cells, but the donor, when you replaced it, radiation is no more there, but it could propagate, but that is a clone of the donor neoblast bearing planaria. This confirmed that the regeneration takes place from the pluripotent stem cell. So this was the first time evidence to show that the entire organism can be produced. Hence, the damaged part also can be produced by the neoblast, which we call them as C. neoblast. Now the question is, what are the questions that remain? Very interesting questions may come from this kind of set of results. First line of questions regarding the clonogenic neoblast. Are all neoblasts pluripotent? If the planaria has got say 1 million neoblasts, are they equal? Or only 100 of those 1 million are so-called C. neoblasts? Because we have evidence to say sometimes into the 6000 radian irradiated planaria, you put one neoblast, it doesn't. Sometimes you put one, it does. So what it tells you, not all C. neoblasts or not all neoblasts are equal. There is some discrepancy, some variation. Data we know already, but we can ask this question. Are all neoblasts are pluripotent? What is the embryonic origin of these clonogenic neoblasts? Are they produced right from the embryonic development, or do they have some other foreign source? What is the hierarchy by which these clonogenic neoblasts generate? 30 or so different cell types of the adult planaria. Planaria has got around 30 different types of cells. Are they capable of making all the 30 cell types? These questions are currently being investigated and with some clues are already obtained. Let us think about some second line of questions regarding the polarity. How does the flatworm cell the anterior blastema to become the head and the posterior blastema to become the tail? How is it possible? How do the blastema derived cells retain their dorsoventral polarity? Dorsal side has got the dorsal structure, ventral side has got the ventral structure. How is it possible? Who is guiding them? So this is some interesting question. Polarity establishment in flatworm. How has it been done? This is something very interesting to follow it up. Flatworms, they are capable of establishing anterior-posterior and dorsoventral polarity. Anterior-posterior means, head to tail, that is anterior-posterior. Dorsoventral polarity means from your backside to the chest. So worm also have got this backside, chest, front side, etc. So two polarities are predominant, anterior-posterior and dorsoventral polarity, using the same factors and the processes employed by the vertebrates. Although the planaria is an invertebrate, one of the primitive line of individuals, but they follow the same strategy, same logic with which a vertebrate is following their anterior-posterior polarity and dorsoventral polarity. This is something very interesting. What is their dorsoventral polarity? Dorsoventral polarity is maintained mainly by the BMP signaling. BMP is bone morphogenetic protein. So BMP is superabundant towards the dorsal side. So we can say BMP signaling maintains the back as the back, and the absence of BMP signaling and the BMP signaling are turned off in the ventral side. First of all, the BMP distribution is high in the dorsal side and the concentration fades towards the ventral side, one thing. And not just that, in the ventral side, you have got the expression of noggins, superabundant. That means even if there are traces of BMPs expressed in the ventral region, the noggin makes sure that they are not going to do any biological function. So noggin, which is an antagonist of BMP signaling, is expressed superabundant in the ventral region and its concentration depletes as it moves towards the dorsal side. You can see in this picture that BMP is very high top and it fades off, and the noggin is very high in the bottom and it fades off. And the BMP expression defines the dorsal region in flatworms. The BMP inhibitors such as noggin are produced in the ventral side. So in a stricter sense, you can say presence of BMP signaling and absence of BMP signaling decides the dorsal and ventral. Presence of BMP signaling, because noggin is an antagonist, it doesn't cause any signaling. So presence of BMP signaling and absence of BMP signaling is enough. Pure presence, pure absence will decide the dorsal and ventral. And there is something we discussed about the mediolateral angle. That means from your chest region to your right hand, chest region to the left hand. So this is your mediolateral angle. That is also decided by noggins. So noggins are expressed right in the center of your median line and your right hand, or median line and the left hand. That is what you are seeing here. So the noggin makes sure which is towards the ventral side, that abundance is between the central portion and the left side, central portion and the right side. So that gradient also is taken care by the noggin, that is mediolateral gradient. Now, if you see the anterior-posterior polarity, anterior most, that is towards your head. Like in this planaria, you can say this is the anterior most portion where its eyes are there. And this is the posterior most portion, that where its tail is there. For you also, your head or neck, that is the anterior most portion, and your buttocks, not your feet, because feet is two structure that is projected from you are basically, your body is only from your mouth till the anus, that is your body. Legs are extension of that body. So from your mouth till your buttocks or your anus. Anus is your posterior most portion. Same logic applies in this planaria body also. Anterior posterior polarity is determined by wind signaling. We have seen the same wind signaling happening in Hydra also, and the same wind signaling is involved. The gradient of wind signaling is involved in the anterior posterior activity, anterior posterior gradient also. Wind is produced in the posterior end. So wind activity is extremely high in the posterior end, and the wind inhibitor is extremely high in the anterior end. So now you have got a gradient, same like what we saw about the BMP signaling. You have got super abundant expression of wind ligand and also the stabilization of beta catenin in the posterior end, and it is fading off as you move towards the anterior end. So what do you understand? If you want head, you don't want to have wind signaling in the head region, or the maintenance of head require absence of wind signaling. And in the anterior most portion, you have got wind inhibitors. Many wind inhibitors are there, like DKK is there, SFRP is there, WIF is there. Many molecules are there which are active inhibitors of wind signaling. They are super active in the anterior most portion. So like we saw in the case of BMP signaling, presence of BMP signaling and absence of BMP signaling. Same way, you have got presence of wind signaling in the posterior and absence of wind signaling in the anterior. There is wind inhibitors do not create a new signaling. Wind inhibitors make sure that wind signaling do not happen here. So that is good enough for getting a head region. So wind antagonists are produced superabundant in the anterior region, so that even if there is a traces of wind signaling or wind ligands or stabilization of beta catenin happens in the anterior end, that is non-welcome. That shouldn't happen. So that is the logic of maintaining the anterior posterior axis. Now, can we think of tweaking this AP polarity during regeneration of the flatworm? So we have seen it. You can take a flatworm and you cut it. You want to take DJ, you want to take SMED, whichever species you want, and you cut it, it grows back. There is a blastema that will be formed. Normally, winds are produced in the posterior blastema. Say, I took a planaria, cut off its head. Like you can see here, it's a normal planaria. I cut off its head here. Then, head has to form. I cut off here, tail region. The tail has to form. This is the normalcy. But Say if I cut the tail, what happens to this rest of the body? I cut here and this piece is gone, and what happens in the rest of the body, the remaining portion? What happens? Winds are produced in the posterior blastema, which then forms a tail bud that will eventually lead to the formation of the proper tail. But if RNA interference is a technique used to block the gene expression. Normally, people use siRNA, people use shRNA. Basically, it means if you are making any given mRNA bind with this siRNA or shRNA, it will be marked for degradation. That RNA will be marked for degradation. Hence, you will not get that protein. So it is as good as that gene knockout. So siRNA, RNA is used to create a gene knockdown, not knockout, because genome has that gene, RNA is being produced, but RNA will never get a chance to make the protein. So RNA is used to target either Wnt or beta-catenin. So Wnt is the ligand. So if you use an RNA against Wnt, it won't be able to go and bind to the frizzled receptor. But if you get rid of the beta-catenin, what happens? Whether Wnt is there or not doesn't matter. The beta-catenin will be not stabilized. Beta-catenin will not get a chance to turn on Wnt signaling specific genes. So you can use either of them. What happens? If you use RNA against Wnt or beta-catenin, the posterior blastema forms a head. This is something very interesting. If you cut it, the tail, you cut the tail here and you blocked Wnt signaling, and instead of getting a tail, you end up getting a head. As you can see here, Wnt is blocked. So this was a normal planaria. Its back is cut, tail was there. All I did was I blocked the Wnt signaling. Now I ended up getting double-headed planaria. So sometimes if beta-catenin is blocked, so in this case, the Wnt is blocked, and in this case, beta-catenin is blocked. So both are giving the same phenotype. Because Wnt is blocked, there is no beta-catenin stabilization. In the second case, beta-catenin never get a chance to go to the nucleus, even if there is Wnt. So that is the only difference. There also, you end up getting a head that is being formed. But it is slightly different. If you note carefully, this looks like a proper head. This do not look like a proper head. If you think a little bit, you will know the answer, because beta-catenin is an essential gene. Beta-catenin is needed for maintaining the cell's integrity. So by knocking it down, it is having a compromised cellular integrity. That is why you don't see this much beautiful head being formed from here. This planaria is fine, but the head doesn't seem like that beautiful. All the eyes are not there. It looks like shrunk. It looks like it is born in a famine period, or it's undernourished. That look is because beta-catenin is not just for going to the nucleus. Beta-catenin is needed for the housekeeping function. That is why the head looks smaller. Now, if you look some more facts on the neoblast. In planarians, the neoblasts are the only cells that divide. If an eye is there, if an intestine is there, if there is a flame cell is there, there is a gamete, neuron, whatnot, there are 30 different cell types are there, but they do not divide. Once a cell is being formed, it is like our neuron. Our neurons normally don't divide, especially the central nervous system. It stays for rest of your lifetime. Many of your muscles, they don't divide. They are terminally differentiated. So planaria, every cell in its body is terminally differentiated, but for the neoblast. They are the only cells that divide. The planarian PV, one special type of planaria, PV homologs are well-known neoblast marker. So PV homologs are a marker for this neoblast, and they have a specific marker that is expressed in them, and they are used to detect them, because you can use the markers of neoblast and you can track them. You can stain for it and then look whether they are incorporating a BRDU or do they have any proliferation marker, etc. So PV is used as one of the target molecule to detect. So PV and the closely related argonauts, other proteins. So argonauts are other group of proteins. They are highly conserved mediators of gene regulation. Like I told you about siRNA, I also told you about shRNA. It goes and binds to the mRNA and it degrades, right? For this, it has to form a RNA-induced silencing complex, RISC, and this is formed with the help of proteins such as argonauts. Okay, so PV and closely related argonauts are highly conserved mediators of gene regulation in general and especially in planaria. Argonauts are widely expressed and mediate gene silencing via small RNA like miRNA and siRNA. This is something important. So we should understand sometimes some animals or some cells do not have adequate amount of argonaut, etc., because they do not want any gene silencing occurring in those cells. But whereas other animals rely a lot on this siRNA and miRNA mediated gene regulation. So this example of this PV and argonaut, it indicates that they are widely expressed in planarian neoblast. One essential function of PV proteins is transposon silencing via PV associated small RNAs. They are also known as piRNAs. So what we understood from now is that the expression of or the quality of genes that are expressed in a differentiated planaria versus the quality of gene expression in a neoblast, they are extremely important to maintain their pluripotency status, to maintain their differentiated status. So that is why they rely a lot on this piRNA, because it is mediated via this RISC complex proteins. So that is why their gene regulations are very stringent. One more thing we need to know about those animals that follow this RNA mediated gene regulation is that they can handle the situation. Like one example I will tell you, many neurons need to have abundant expression of LET7 microRNA. LET7 microRNA, they target lot of mitotic mRNA, genes that are capable of driving the neuron into a proliferative phase, and you don't want that because neurons are our storehouse of information. A lot of vital biological functions are there. So LET7 microRNA make sure that even by accident a given promitotic genes are turned on, the cell will never think of dividing its DNA, because LET7 make sure that that newly formed mRNA will never get a chance to get translated. In other words, microRNAs can act as guardians of maintaining their status. Same way, a neoblast need a particular status. It should allow, if the gene expression events, then another set of gene regulation events will kickstart. So understand, all the planaria is a simple structure. The gene expression events in a given cell decides whether it is terminally differentiated or the neoblast need to be constantly under their proliferative phase. So this is what we should keep in mind, that piRNA is very important in regulating their gene expression. Transposons have been estimated to account for 31% of SMED, Smithia mediterranea genome. 31% of their genome, that means 1/3 of their genome is having transposons. What does it tell you? It has the vulnerability to reshuffle, rearrange their genome. Transposons are jumping genes. That means they have a transposase coded in them. They can use the transposase and cut off from that region where they are there. They can insert into a new place. That means the genome is vulnerable for alteration. These are all some more facts. What we learned is some more additional facts about the neoblast, because neoblast, having said that, they have the great capacity to regenerate lost tissue part. We should also understand the underlying gene expression or the underlying gene regulation events are pivotal in controlling these remarkable properties. We will study more about regeneration in planaria in the next class. Thank you.